Hello, everyone. Thank you for joining us for our third presenter. Liang Zhao from the FDA will be discussing model-informed drug development, model-integrated evidence to support generic drug development and assessment. I'd like to take it to you, Liang Zhao.
Good afternoon, everyone. Nice to see you here, my colleagues, my friends. I see many familiar names. Thank you for your time and interest. Today, I'm going to talk on using model-informed drug development and model-integrated evidence to support generic drug development and assessment. So for quantitative method modeling are not only technical tool set, but also quantitative accurate summary of information and knowledge throughout a product life cycle. Quantitative method modeling provides a critical knowledge base for regulatory decision makings for all subject areas. Ryan, if you keep moving, so sorry for the animations. The QMM provided critical knowledge base for regulatory decisions for all subject areas, including in vitro bioequivalence method, drug device combination products, post-marketing surveillance, and in vivo B method for generic drug development and review. By taking advantage of prior information that have been accumulated and characterized by models, we can use model-generated data as the pivotal information for drug approval. It's more like represent one step further from MIDD. Here, we are talking about using model-generated data for pivotal information for drug approval, rather than to inform drug development and design. I will illustrate the point in the following slide. Next one. In this diagram, if you keep moving, right? In the diagram, the inner circle shows the QMM tool set. The bullet points outside the circle represent their corresponding impacts. The current tool set, including QCP, Pvpk, modeling, and data analytics, including big data analytics. Data and big data analytics are the area undergoing rapid expansion in their utilities in regulatory science. We have pioneered in this area by publishing several original works in the field. However, this is going to be out of the scope of today's talk. Next slide. Modeling simulation impacts various regulatory activities in the Office of Generic Drugs. In calendar year 2019, in 2020, we have similar picture. QMM-related regulatory review activities include ANDA reviews, pre-ANDA meetings, and the guidance development. For pre-ANDA meetings, it's more resemble the pre-IND and a phase 2 and a pre-NDA submission meetings for new drug development. Modeling simulation-related regulatory scientific activities include both internal and external researches. The external researches are conducted in the form of contracts grant. We have we have almost collaborations with all the well-known labs across the globe in terms of research collaboration. Next slide. One of the frequently applied modeling tool set is certainly quantitative clinical pharmacology, or you can rename it as pharmacometrics. QCP has been widely used for BE study design and data analysis to support science-based BE recommendations. Bioequivalence can be evaluated using PK endpoint, PD endpoint, and clinical endpoint. When using PK endpoints, sparse PK sampling, endogenous baseline corrections, and long study durations for long-acting injectables can pose challenges in practice and assessment. Model-informed or model-integrated approaches serve as the very tool set to optimize study design, evaluate B metrics, and assessment of B acceptance criteria, and reduce study duration and sample size. When using PD endpoints, QCP is used to guide computational data analysis endpoint identification and selection, and optimization of study design. So I'm going to skip the rest of the slide because today's talk is mainly focusing on the Pvpk applications. Next slide. Here, but I still want to highlight some of the QCP regulatory impact in 2019. For B study design, I just want to mention that we finally published the product-specific guidance for the levonorgestrel intrauterine system in January 2020. The progestin containing IUS was approved in the United States for intrauterine contraception for up to 5 years. 5 years, you can imagine what that states for. However, a 5-year clinical study may not be realistic for B evaluation. A modeling work was conducted to reduce the B study duration to 1 year and to establish the corresponding B acceptance limit that can be used as a surrogate for the B limit at the end of 5 years. We have also launched original modeling work for long-acting injectables, which represent a viable commercial area for industry. The work can potentially reduce the high regulatory burden due to the long study duration again, and the large sample size as a result of a high variability in PK parameters. QCP modeling can also guide the identification of clinically relevant PK metrics, such as partial AOC and PD metrics that are more sensitive to drug exposure at the site of action. It also have good B2, 505B2 new drug implications. Similarly, in the event of aberrant observations and a Tmax shift, QCP modeling simulation can be employed to assess the associated clinical performance. Next slide. So this is going to be the focus of today's talk about another side of a tool set, which is the Pvpk modeling simulation. First, Pvpk has been resorted by industry to support the in vivo portion of a generic drug product development program. I think the former portion is about a new drug as well, how to use the Pvpk model to support the space determination for in vitro testings. Second, Pvpk modeling is a key tool set in advancing in vitro and in vitro or in vitro only B approaches, especially for locally acting products. It can be used to determine appropriate B metrics on systemic PK to ensure local equivalence, to conduct virtual B simulations on local drug exposure based on directly formulation inputs, to identify formulation quality attributes with clinical relevance under the associated B space, to justify small Q3 differences in quality attribute between test and the reference, to guide selection of clinically relevant in vivo B studies. Next slide, please. Out of the Pvpk highlight in year 2019, I would like to specifically mention the FDA approval of the first generic drug product for diclofenac topical gel, where a virtual BE simulation leveraging dermal Pvpk model was performed instead of conducting a comparative clinical endpoint study as recommended in the current draft product-specific guidance. You can immediately appreciate the cost and time saving by leveraging modeling approach in generic drug approval. Novel approaches are being developed in the realm of inhalation and ophthalmic products, where computational fluid dynamics models in combination with the Pvpk models will significantly provide weaponaries to modernize the regulatory science and practice. Of note, modeling advance in these areas will simultaneously modernize the toolset for new drug development as well. Next slide. This is a summary on what quantitative method modeling can do for complex generic drug development and approval in the near future. For complex products, we would work more to allow models to support development of in vitro or in vitro only B method. Under the identification for B spaces associated with this method. We also should work together to allow models to provide the justification of using alternative metrics to replace comparative pharmacodynamics or clinical endpoint studies. We should also work together more on the building platform for virtual simulations. I think that's going to represent the future in the modeling field, how to conduct virtual studies, virtual clinical trials. Certainly, having said that, sufficiently model verification validation will be the key to assess the regulatory impact of a model. I kind of heard about the question to the previous presenter regarding model verification validation, and it is really depending on the purpose and the regulatory impact of the model in terms of the extent of model verification validation. Next slide, please. For non-complex products, we need to work together on using quantitative method modeling to expand the scope of BCS3 biowaivers. One practical question to address is when and how non-Q1, Q2 product can be qualified for BCS3 biowaiver. Biowaiver means you don't need to conduct in vivo PK study based on the formulation characteristics and attribute. You can directly bring the drug on the market. We also need to work together on to use how to use modeling to support not contacting B studies for lower strength formulations under special conditions. For example, when the formulation are not compositionally proportional. Although not listed here, it is also a time for us to leverage Pvpk models to predict the food effects on PK and assess the necessity of conducting fat B studies as a situation arises when sufficiently verified and validated model can predict the food effect. In addition, the modeling can be used to extrapolate the B study result from healthy volunteers to disease population and predict the pediatric and geriatric population performance as well. Next slide. So in my following presentations, I will use 3 cases to illustrate the utility of Pvpk models in the regulatory setting. Next slide. The case 1 is about we have developed a Pvpk model for putative BCS class 1-3 drug, oseltamivir phosphate, OP, and its metabolite, OC, in both adults and pediatrics. Then conduct virtual BE simulations to establish BE dissolution safe space for the parent drug in both adults and pediatrics. Next slide. This is the flowchart of the development and verification validation of both the parent and metabolite drug Pvpk model. Here we would like to mention that the validation part, which is very important for Pvpk model building for generic drugs. We validated the Pvpk model using in vitro dissolution data and in vivo PK profiles for both the parent and metabolite following administration of different dosing regimen of the reference and the different generic drugs in adults and pediatrics. For details, you can refer to the recent publication in the AAPS journal as listed at the bottom of the slide. Next one, please. This slide shows the established model could capture the PK profiles of both parents and metabolite in adults following different dosing regimen. The model can also capture the PK of both parents and metabolite following oral administration of parent drug in adolescents and neonates. The next slide, please. The validated parent drug model was used to do the virtual B simulations in different age populations and demonstrated that a different dissolution safe space should be set for different age groups. For example, figure A on the top left shows that in adults to maintain bioequivalence, test a product with a 10% of slower dissolution profile would maintain BE with the reference product. And as shown in figure B, the test product with 12% slower dissolution would fail to meet BE criteria. While as shown in figure C and figure E, a more stringent dissolution boundary was observed for pediatrics, 6% slower dissolution boundary for adolescents, and a 4% slower boundary for neonates. Next one, please. Here is the summary of the first example. Beyond the 2 points I just mentioned, different safe space for different age populations. The case example also highlights the utility of a Pvpk absorption modeling simulation in prediction of BE and provide a quantitative basis for setting clinically relevant specifications for dissolution in both age groups. Next slide, please. In the case 2 is about identify the critical quality attribute for ophthalmic formulation using modeling approach. I'm going to show you 2 of our internal case studies. The first one is ophthalmic emulsion, and the second one is ophthalmic suspension. The first case study is to use tear film modeling to study the effect of cyclosporin ophthalmic emulsion formulation properties such as viscosity, surface tension, osmolality on drug bioavailability, and a tear film breakup time, TBUT. TBUT is a metric that is commonly used as an indirect clinical endpoint in vivo measure for the evaluation of dry eye syndrome. Once applied, emulsion mixes with the tear film, and then oily globule break down and attach to the lipid layer of the tear film. A mathematical TBUT model was developed to predict TBUT as a function of certain critical quality attributes such as non-Newtonian viscosity, osmolality, and surface tension. All parameter sensitivity analysis demonstrated that bioavailability and the change in TBUT, the clinical endpoint, were both very sensitive to viscosity, slightly sensitive to surface tension, and insensitive to osmolality. Currently, OGD has funded an external research project with absorption systems, which uses optical coherence tomography to measure tear film and tear film manifold thickness in rabbits. This ongoing measurement will allow for model validation of the development of cyclosporin emulsion model. The next slide, please. The second case study is dexamethasone ophthalmic suspension formulation. After topical installation of ophthalmic suspension to the eyes, less than 5% of the instilled drug may usually be absorbed into the eye through cornea or conjunctiva tissues. While most of the solid particles and dissolved drugs are cleared from eye surface through nasolacrimal drainage, which will then go through the systemic circulation pathway. In order to understand the ocular and systemic bioavailability of ophthalmic dex suspensions, FDA has recently conducted an internal rapid study with PK sampling from multiple ocular tissues and plasma. An ocular Pvpk model was developed to study the drug absorption and distribution after topical installation of dex suspensions. The development of the OCAT model was verified using multiple data sets, including the particle size impact on ocular exposure, viscosity impacts on ocular exposure, and the nonlinear dose exposure following the administration of different suspension strength. In this case, we do find that the systemic drug circulation may not reflect the local drug exposure at the site of action. Next slide. Can you keep pushing, Ryan? Yeah, thank you. In order to identify the critical quality attributes for ophthalmic suspensions. Parameter sensitivity analysis was performed using the verified DAX-OCAD model. The impact of a drug product strength, particle size, and viscosity on in vivo ocular exposure for ophthalmic suspensions in rabbits was investigated. The parameter sensitivity analysis demonstrated that DAX ocular exposure is very sensitive to the viscosity of the suspension formulation. For example, the ocular exposure increases with the increase of viscosity, which is intuitively understandable. The parameter sensitivity analysis also indicated that the plasma PK is not sensitive to the change in both particle size viscosity of the formulation compared to the ocular PK. Given the time, I will skip the rest of the findings. Please, if you have interest, you can appreciate the result with the shared slide. Next slide, please. So the summary of the K3, I want to, given the time, you can read, and all the details is self-explanatory. Next one. So we have a dermal Pvpk model for diclofenac sodium topical gel, and the Pvpk modeling results have led to the directly led to the drug approval. Given the time again, I will let's keep rolling the slide. So we validated the model with different platforms, and also with different drug product of similar nature was used to qualify the platform, and all these things taken together have led to the drug approval. Next slide, please. Next slide. Next slide. Pvpk model, conclusion slide, please. The conclusion is that modeling simulation has an important role in supporting generic drugs review and approval. We have a publication differentiates what is model integrated evidence and what is MIDD. And that being published in 2019 CPT journal. And we have also various meetings and publication in terms of model verification and validation for Pvpk model to impact regulatory decision making. Looking in the future, I just want to give you a heads up that we welcome more collaborations among the agency, the academia, and the generic industry, because your collaboration are key to the successful value creation for generic drug and a new drug development and approval for a quantitative method of modeling. And in June this year, we are going to have FDA public workshop to solicit the public input regarding where are the knowledge gap, where we can do further in terms of research to advance the field, to make a model more useful and more impactful in terms of regulatory decision making. With that, I want to thank many of my colleagues in the acknowledgement slide and all the modelers in the field, our contractors, grantees, to be able to make the presentation possible. Thank you.
Thank you. We've got some time for questions as they come in. Thank you very much, Yang Zhao, for the wonderful discussion. One of the questions I have for you is early on, you've got a slide kind of summarizing a wide variety of how QSP efforts have, physically and very strongly impacted, various guidances and approaches that the OGD has taken for drugs. One of the questions I have for you is when that modeling efforts come through, is it typically handled through internal modeling exercises at the FDA, or is it typically through sponsors submitting ANDAs that are looking to drive changes within the ANDA programs that they're interested in?
Yeah, Ron, that's a great question. And there's a distinction between new drug and generic drug development. I think in the new drug, it's mainly the modeling advance mainly originated from the industry. For generic drugs, given the nature of the industry, with one reference, there are multiple competitors targeting the same product. There's a barrier in terms of sharing. So, and also the pocket is very shallow in the generic drug industry, but there are multiple, many firms. Then the model is that we collect the user fee, and then we allocate a small portion of the user fee to allow research within FDA. We can also conduct research through collaborations with external experts where grants on contract. That's how we are doing it.
Excellent. Along the same lines, I know due to time you had to kind of go through it a bit, but on your Pvpk model, I noticed that when you validated it, the validation was actually greatly improved by optimization there at the agency. When you're receiving a submission, do you give any more weight onto whether that kind of steps are done by the sponsor versus done internally?
Yeah, we should primarily rely on the applicant or sponsor to conduct the model verification validation. I should say it's the state of art. It's also risk-based. We are not applying very stringent area for every type of the model. It's totally depending on the use. Also, we are reasonable group, and we do weigh in all the other totality of information to evaluate. Okay, even think about it. Even for physiological parameter, people can win a Nobel Prize to figure out what exactly the value is. Yeah. But depending on the purpose, we may not need to figure out all those nitty-gritty details. If it fits the purpose, then we'll take the model.
Yeah, makes sense. Let's see if there are questions coming through. So one of the things that I know a lot of generic companies are really excited about is the idea of using modeling in lieu of conducting like a PKBE study. I know there's a couple examples like the diclofenac one you presented. Do you think that that's going to occur more and more in the future? Or additionally, is there a way that a generic drug sponsor could tailor their modeling package to give you more confidence in its ability to stand in place of clinical studies?
That's a great question. That's one of the initiatives we are trying to push in the agency. We really want to exemplify the impact on the strategic side of the modeling community. We are not technicians. We, as I mentioned at the very beginning, modeling is a quantitative summary of all the knowledge and data gained in the drug development. We are going to do more and more in the future. I think generic drug, the place will be a start. The advancement in the generic area will be even faster than the new drug side, given that we have already learned a good deal about the drug efficacy, safety. Now we want to leverage that information into the formulation assessment and into the PK exposure differences, and we have a good impact to make in this field. I think hopefully the impact we make in the generic field can spill back to the new drug side, in terms of B2 applications. And some techniques we developed, for example, the computational fluid dynamics coupling with the Pvpk model can also have a good, offer good insight in the new drug development for already inhaled drug product or intranasal drug product. And for skin topical product, we also have developed a good deal of modeling research, and that have a very tight connection with the real world.
Thank you again very much, Zhao, for taking the time to join us. Again, I encourage everyone to go ahead and leave the session and join the next one. We will be talking with Gustavo Mendez Lima Santos. Thank you very much, everybody.
Thank you very much all.